In Lab 2, we're going to be replicating some of the analyses from this paper by Race et al. So to get started, let's talk about this Excel sheet, Excel questions. It has 4 different tabs, and we'll talk about each of those. So in this first tab, this is essentially the data from the experiments looking at the sentence superiority effect. So whether or not people are better able to remember sequences of words when they're in sentences versus random lists. So here we first have our control participants and their data. In each of these columns, we have their proportion accuracy for different word lengths. So this is 3-word sentences up to 8. And then here we have the data for the random lists, starting with 3 words up to 8. And so here, for example, this person got 100% correct for the 3-word sentences as well as the 3-word lists. And over here we have the magnitude of the effect, which is just sentence minus list accuracy. So here you can see there's no difference because they did the same on those. Over here we are computing an average, and if you go up here and you click in this formula bar, it will show you the cells that it's using. So here we can see that it's computing the average of the effect for the first control participant. Down here we have the same kind of data, but for our patients with medial temporal lobe damage. And here we are further categorizing these patients based on whether they have damage that extends beyond the hippocampus, denoted by H+, or only limited to the hippocampus, denoted by H. Again, we have the accuracies, magnitude effect, and average for each participant. And down here, this is going to be averaged across the different groups. So if we click on this cell here, we can see we're computing another average. And if we click up here, we can see that it's computing the average for all of the control participants when they're looking at 3-word lists. Okay, and so each of these will compute the average for different conditions. And over here, this is computing the standard error for each of those conditions as well. Here we have our figures, and if you click on the bars, it'll show you what data is being used. And then again, more means, but separating the subgroups for the participants with only hippocampus damage or hippocampus plus more damage. Experiment 2 is looking at the visual bootstrapping effect. So experiment 2A is just confirming that participants are faster when using a typical versus non-typical keypad, just to make sure that everyone is equally familiar. These numbers are log transformed reaction times, and you don't need to worry too much about what that means, except that higher numbers are going to be slower. And so we can see that in general, everyone is slower for the atypical keypad, as we would expect. Then experiment 2B is actually testing that bootstrapping effect. So here we're seeing if when people are visually presented with these numbers in either typical or atypical keypad formations, are they better at remembering those sequences of digits? Okay, and so now over here, these numbers represent proportion accuracy for controls as well as patients. This third tab here is showing the same data as was in the first tab, but just in a format that we can use for RStudio. And then this last tab, again, is the same as the data shown in experiment 2, but formatted for RStudio. So now let's move on to RStudio. So here I have the race.r script from last time. For this lab, you're going to want to re-download the race.r zip file, unzip it, and start fresh. So that's what I've done here. First, I'm going to start by highlighting these lines, which are loading some packages that we'll need for next steps. So I'm going to highlight those and then hit Command Enter to run them, or you could hit the Run button up here. Next, I'm going to read in the data that we need, and then I'm going to run our first ANOVA test. So what's going on here is we're using this ANOVA test function. We're telling it what data to look and we're also giving it our dependent variable dv, which is the proportion correct. So what we're really testing for here is we want to see how accuracy varies depending on a couple different factors. One is whether or not they have damage to the MTL. We also want to look at how accuracy is affected by sentence length, and also how it is affected by the context, whether the words are presented in sentences or lists. Okay, but we need to label these variables depending on how they vary between or within people. So the group variable, which is whether these are healthy controls or amnesic patients, is a between variable because it varies between people. You can only be one or the other. And then our length and context variables are within subject variables because they vary within people. In other words, every participant is shown multiple trials with different lengths and different contexts. So we're going to go ahead and run this all together, hit Command Enter, and take a look at the output. So here is the output, and each of these lines is showing the significance of different effects. So in the first line, we have the main effect of group. So this is asking whether or not having damage to the MTL is going to affect your accuracy. And the answer is yes, because here we can see our p-value is less than 0.05, meaning it's significant. You can also tell by the asterisk, which means that the result is significant because the p-value is less than 0.05. And just to go through each of these rows, we have our the effect of interest, we have the numerator, degrees of freedom, denominator, degrees of freedom, F statistic, p-value, whether or not the p is less than 0.05, and then GES is a measure of effect size. When you have these rows that have the colons, that means it's some kind of interaction. So this one is an interaction between group and sentence length, or list length. So in other words, does the effect of list length depend on whether or not you have MTL damage? For example, maybe if you have damage, then you're going to have a steeper decrease in accuracy. If there's no interaction, then you would see similar slopes across the different list lengths for both groups. But here we can see again it is significant, meaning that the effect of list length does in fact depend on what group you belong to.